This is the type of microscope you will be using in the lab. It plugs into an electrical socket, and sometimes there is a power switch at the back. The brightness wheel is here. It controls the bulb, which is here. The bulb is the only electrical component of the microscope. It acts as a light source to illuminate the specimen from beneath. Sliding the brightness wheel controls the brightness of the light. There is a lens here called the condenser. This focuses the light onto the specimen. This platform is called the stage. The metal clip holds the slide in place. Using the controls here, the slide can be moved forward and backwards, or left and right. The objective lenses are mounted on a revolving turret. Each objective lens can click into place, like so. The magnifications are written on the sides of the objective lenses: 4x, 10x, 40x and 100x. The 100x is an oil immersion lens, which means that a drop of oil must be placed on the specimen before the lens can be used. The immersion oil has the same refractive index as the glass inside the lenses, which means that as much light as possible will enter the lens and the resolution will be as high as possible. All the other lenses are called dry lenses, and it is absolutely critical that they never come in contact with oil. Immersion oil can cause severe damage to a dry lens. At the top of the microscope, there are two eyepiece lenses. These magnify the image by a further factor of 10. The interpupillary distance can be adjusted by sliding the eyepieces closer together or further apart. The focus of the microscope image is adjusted using the large coarse focus wheel and the smaller fine focus wheel. These wheels are accessible from either side of the microscope, and they function simply by raising or lowering the stage. Having adjusted the focus so that the image is clear in the right eyepiece, the diopter ring on the left eyepiece may need to be turned so that the image is equally clear in the left eyepiece. Sometimes adjusting the focus is not enough to make the image clear. It may also be necessary to adjust the amount of light reaching the specimen. This is achieved by opening or closing the iris diaphragm using this lever.